So some general functions of TNF, IL-6, and IL-1 to help make that past slide a little bit less overwhelming. It acts on the bone endothelium to release neutrophils. You may recall from your development lectures that neutrophils, like many hematopoietic cells, will arise from the bone marrow. When more are needed, the bone marrow will activate the production of neutrophils that can then go out into circulation and migrate to the site of infection thanks to chemokines like CXCL8. TNF, IL-6, and IL-1 also act as pyrogens. Remember, pyrogens increase the body temperature of the host through the production of prostaglandin E2. These types of cytokines are also thought to signal on the hypothalamus, fat cells or adipocytes, and muscle cells to mediate changes in energy usage, thus making the host more unfavorable for the pathogen. You may learn later on that many of these energy changes are also important for promoting tolerance in the organism. This means that as the pathogen and the immune response cause damage to the host will be more able to tolerate that damage and survive the long-term effects of having been infected. The important thing to know about these immediate response or innate cytokines is that if they're overstimulated or overactivated, they can actually become detrimental to the host and the immune response. Normally, what should occur, and we're going to use TNF as an example here, is that when you have a local infection with, say, a gram-negative bacteria, macrophages in that local area will begin to secrete TNF-alpha to tell the rest of the body that there is a local infection, in this case in the arm. This will cause the release of plasma proteins that will act on blood vessels and nearby endothelium to open them up and be a little bit more permeable. This permeability of blood vessel walls will allow the migration of immune cells into the site of infection. Here, you get phagocytosis of bacteria, local vessel occlusion, and plasma cells and T cells draining into the local lymph nodes to generate a local immune response that will ultimately benefit the host and help clearing the pathogen or the infection. However, if you get systemic activation of macrophages to produce TNF, or macrophages in circulatory sites like the liver or the spleen become activated to induce TNF, you can actually result in systemic-wide increased permeability of blood vessels, causing severe edema, decreased blood volume, neutropenia followed by neutrophilia, as well as the collapse of blood vessels as all of the fluid leaks out of the blood vessels due to the increased permeability. All of these changes can be incredibly detrimental to the host and cause disseminated intravascular coagulation, which will result to wasting of the host and multiple organ failures due to loss of blood supply and thus oxygen supply to those particular organs. This is known as sepsis and is an increasingly and also continuously fatal problem in the clinic. So what do TNF, IL-6, and IL-1 actually do to benefit the host in addition to increasing the permeability of blood vessels once they're induced? Well, they also participate in this thing called the acute phase response. In the acute phase response, TNF, IL-6, and IL-1 act on liver cells or hepatocytes, changing the profile or the proteins that they're secreting into circulation. These new proteins that are produced by the liver in response to IL-6, 1, and TNF are called acute phase proteins, and they have many functions. Acute phase proteins are similar to antibodies in that they have a broad specificity for pathogens and only need cytokine activity to be released. This means that they're not antigen specific and they won't respond in a pathogen specific way when the body's been infected. Some good examples of acute phase proteins include CRP or C-reactive protein. CRP is a pentraxin or a 5 subunit molecule that binds and opsonizes bacteria. Another protein produced by the liver when it's been induced to activate this acute phase response is mannose binding lectin. Mannose binding lectin is an important component in opsonizing bacteria and activating complement. This is something that you'll hear a little bit more about in a different lecture. Essentially, the role of the acute phase response is to initiate the destruction of bacteria by coating it with molecules that will tell macrophages, hey, this is the bad guy, you need to come and eat this now and destroy it. Much of what I just talked about were cytokines of the innate immune response. However, what do cytokines of the adaptive immune response do? Because they're very much present and they are very important in maintaining an adaptive immune response. Well, one example is IL-7. IL-7 is important not only in the actual mounting of the immune response, but also in the production or the establishment of the immune response early on in development. IL-7 is necessary for the survival of B and T lymphocytes as they develop. You can see here in the bone marrow, you have this multipotent progenitor cell that then differentiates into this common lymphoid progenitor, meaning that eventually it can become a B cell and stay in the bone marrow, or make its way towards T cell differentiation by migrating up to the thymus. However, in order for the lymphocyte to proceed beyond this stage, it needs to receive signaling via IL-7. IL-7 will tell the cell, you're in a good place, the environment is good, we're ready for you to develop into appropriate lymphocytes now. IL-7 is still important later on. As T cells circulate through your body in their naive form, they'll always circulate back through lymph nodes in the spleen where they can receive more IL-7 signaling to tell them, you're still young, you can still survive a bit longer, and look for any invading pathogens that you may be specific for. Another example of a more adaptive immune cytokine is IL-2. IL-2 is very important for proliferation and differentiation of T cells. However, the T cell is always receiving some IL-2 signaling to let it know that it can survive, similar to IL-7, and this signaling increases drastically when the T cell becomes activated. You can see in a naive T cell that they still express the IL-2 receptor, but it's a receptor with moderate affinity, which means that the full signaling activation of IL-2 doesn't occur. However, when a T cell becomes activated, it upregulates this third component of the receptor, the alpha component, allowing it to become a high affinity receptor and respond much better to IL-2 signaling. This is a more schematic example of that here. As we talked about, resting or naive T cells will express a moderate affinity IL-2 receptor, consisting only of the beta and gamma chains. However, when they become activated, they'll express all three receptors, alpha, beta, gamma, and begin to secrete IL-2 themselves in response to receiving that first IL-2 stimulus. The binding of IL-2 to its receptor in both a paracrine and an autocrine manner will cause accelerated cell cycling of the T cell. This also modulates T cell differentiation and induces and enhances proliferation in response to having been activated. This means that once a T cell sees an antigen and receives all of the necessary co-stimulatory signals, it will begin to rapidly proliferate in order to mount a very highly specific and effective response against the invading pathogen. Another important role for cytokines in the adaptive immune system is something that you should already be very familiar with. This is signal 3 of activation of T cells. As you may recall from other lectures, T cells need to receive 3 separate signals from the APC in order to become activated. The first of which is the TCR-MHC interaction made in an antigen-specific manner. However, that's not enough. The T cells also need to receive signal 2 through CD28 and B71 and B72, or CD80 and 86. And finally, they also need to receive this third signal, which is cytokines pumped out from the APC to tell the T cell, now you're activated, this is the type of immune response that you need to mount, or this is the type of T cell that you need to become. You may recall that interferon gamma and IL-12 are important for differentiating T cells into Th1s. IL-4 is necessary for Th2 differentiation, TGF-beta, IL-6, and IL-23, important for Th17, IL-6 for T follicular helper cells, and TGF-beta and IL-2 for induced T regulatory cells. It's also important to note that cytokines of the adaptive immune system, in addition to being fate-specifying, are also very important for effector function. Even after these T cells have differentiated into their specific subtype, they'll still continue to secrete different types of cytokines, including gamma, IL-4, 5, and 13, IL-17, and IL-10 and TGF-beta to mediate their appropriate effector response. So what about chemokines? Because cytokines that don't cause migration of T cells aren't the only important signaling molecules in the adaptive immune response. Chemokines are also important. As I mentioned earlier, please recall that chemokines are produced by all different types of cells and are necessary for guiding immune cells where they need to be in different types of infections. However, they're also important for establishing and maintaining the immune response, similar to IL-7. Chemokines are actually necessary for organizing the lymph nodes and the structure of the lymph nodes. We won't go through this schematic panel by panel, but what's important to know is that the migration of T and B cells to their appropriate sites within the lymph node is highly dependent on these chemokines. CCL21 is necessary for driving cells into the lymph node itself. CCL19 also helps for the migration of T cells to the T cell zone within the lymph node. And CXCL13 is responsible for attracting B cells into the B cell follicle. You'll also learn that CXCL13 is a necessary cytokine in establishing the germinal center after B cells have become activated. This is a good way to show that chemokines are not only important once the infection has been sensed, but also important in maintaining the general integrity of the immune response. To that note, it's also important to remember that chemokines are essential for integrating the adaptive and innate immune responses. Many of these cytokines will signal through the same receptor, and many of these receptors are expressed on different cell types, but at different times, which will help orchestrate an effective and highly organized immune response to varying different types of infections. This brings us to our last objective for today, which is to briefly introduce interferons. These are personally my favorite type of cytokine, so I look forward to this last objective. Interferons are also cytokines, but they're named because of their ability to interfere with viral replication. There are three subtypes of type 1 interferons. Type 1 interferons, which include interferon alpha, beta, kappa, epsilon, and omega. There's also the type 2 interferon, of which there is only one, interferon gamma. And there's a third family of interferons, which we'll talk about not at all today, but are still interesting. And this includes interferon lambda, which is composed of IL-28 alpha, beta, and IL-29. So the three different families of interferons actually have very different functions and are produced by different cells at different times to mediate different functions. Type 1 interferons, which are my actual favorite, are produced by specialized immune cells and not by all types of cells. So while many cells are capable of producing type 1 interferons, the real professional producers of type 1 interferons are plasmacytoid dendritic cells, or PDCs. They play a large role in producing massive amounts of type 1 interferon when the organism has been infected with the virus. They do this by expressing a subset of TLRs, or toll-like receptors, and other sensors that recognize viral PAMPs, or pathogen-associated molecular patterns, and this in turn will activate type 1 interferon production. So you can see here, most of the time viruses are recognized due to their RNA or DNA. This is something that you may have heard about or will hear about in other lectures. But this recognition of RNA or DNA by the plasmacytoid dendritic cell through TLR7 and 9 respectively will allow for the massive increase of type 1 interferon production, including interferon alpha and interferon beta. These type 1 interferons also use a JAK-STAT signaling pathway to transduce signals, similar to the other cytokine receptors or the tyrosine receptor kinases that we talked about earlier. Type 1 interferons signal through a very specific type of receptor called IFNAR, or interferon alpha receptor. Downstream of IFNAR, the dimerization of the two IFNAR subunits is JAK2 and TYK2. These, more often than not, will lead to the phosphorylation of the transcription factors STAT1 and STAT2. Many other STAT transcription factors can actually also act downstream of type 1 interferons, but their function is less canonical and also less well characterized. Once STAT1 and STAT2 have been phosphorylated, they can interact with this molecule or this other transcription factor called IRF9. Together, these three components will form a complex that we refer to as ISGF3 over there. It is ISGF3 itself that is able to migrate into the nucleus and drive the transcription of a whole subset of genes, of which there are hundreds, called interferon-stimulated genes. All of these interferon-stimulated genes have the general same goal, which is to interfere with viral replication. This is a bit of a flow chart showing some of which I just said, but also showing some of the downstream molecules that are produced as a result of type 1 interferon signaling. These are all within this interferon-stimulated gene family and include MX proteins, 2 to 5' linked adenosine oligomers, and the kinase PKR, all of which are responsible for interfering with viral replication. ISGs also include IFIT proteins, which suppress the translation of viral RNA. ISGs can also result in the upregulation of MHC class I expression, which will increase antigen presentation on infected cells or cells that have the potential of being infected. ISGs will also result in the activation of dendritic cells and macrophages. They activate NK cells to kill viral infected cells. And they induce chemokines to recruit lymphocytes. So as you can see here, the induction of a type 1 interferon response is essential for appropriately responding to viral infection and is oftentimes the turning point in whether an organism can successfully clear and respond to a viral infection. An example of one of these antiviral proteins, or these ISGs, are IFITs. So normally what will happen in a cell is you have your tRNA, your 40S ribosome subunit, and then EIF2, 3, and 4, which all assemble to form a pre-initiation complex, which will then allow for the induction of translation. This complex then joins up with the 60S ribosome subunit and releases EIF2, 3, and 4. The release of EIF2, 3, and 4 actually activates the translational activity of the ribosome and thus the production of proteins. You'll remember that as viruses infect cells, they oftentimes will utilize this host machinery in order to translate their own proteins and thus build up more virus and replicate. However, with the induction of IFITs, this complex, or the pre-initiation complex, can't form appropriately and thus stops the translation of all proteins, including viral proteins, in an infected cell. Type 2 interferons are a little bit different than type 1 interferons, and the one type 2 interferon, interferon gamma, has many roles. A lot of these you've already heard about before in various lectures, but I'm going to try and sum them up a little bit in the next few slides. So you can see that interferon gamma can be produced by a few different subtypes, including Th1 cells, T follicular helper cells, and cytotoxic lymphocytes. These are CD8 T cells. And they can act on many different immune cells as well. All of these immune cells, when they receive interferon gamma signaling, will enhance their inflammatory or immune function and aid in the clearance of the pathogen. Perhaps one of the most notable functions of interferon gamma is this activation of macrophages. So you can see here that when a Th1 cell recognizes an infected macrophage or even an uninfected macrophage, the production of interferon gamma by Th1 cells will activate the macrophage to become more microbicidal. This means that the macrophage will upregulate the proteins it needs to properly perform phagocytosis and just simply become better at killing invading pathogens. Interferon gamma, once it activates macrophages or dendritic cells, can actually also feed back on the producer cell itself. This means that interferon gamma, after having signaled through the interferon gamma receptor on APCs like macrophages and dendritic cells, can then drive the transcription and expression of different cytokines that then become helpful for the initial cell that produced it, or for other cells that are necessary to respond to the specific infection. So for example, if you have interferon gamma signaling through macrophages, you can drive the transcription of IL-12 to induce more Th1 cells, or the transcription and expression of IL-23 to induce Th17 cells, which would be necessary for different types of infections like parasites. So to finish up for today, let's revisit some of our objectives and the main points that you can take away from today's lecture. So in our first objective, where we defined cytokines and learned about the different families and receptors, we learned that cytokines are small signaling proteins that can act in an autocrine on themselves, paracrine on their neighbors, or endocrine everywhere else manner. Cytokines are part of 4 different families: the IL-1 family, the hematopoietic family, the TNF, and the interferon family, that all use different types of receptors to signal. We also learned that the majority of these cytokines signal through this JAK-STAT signaling pathway, which results in the transcription of genes specific for the response that the body requires. In our second objective, we defined chemokine and distinguished between the 2 types of chemokines, CC and CXC chemokines. We learned that a chemokine is just a subset of cytokines that directs the migration of immune cells. We now know that CC chemokines have 2 adjacent cysteines at the N-terminus, whereas CXC cytokines have 1 additional amino acid in between those 2 cysteines at the N-terminus. We know that CC cytokines recruit monocytes, lymphocytes, and other immune cells, while CXC chemokines are very good at recruiting neutrophils. We also know that chemokines signal through a different type of receptor called a GPCR that uses G proteins to transduce the final function. In our third objective, we strove to understand the importance of cytokines and chemokines in mounting an inflammatory response. We learned about 3 very important innate cytokines, TNF, IL-6, and IL-1, that are important for mounting the initial inflammatory response. We learned about how they mobilize neutrophils from the bone marrow and increase the temperature of the host, and also how they signal on the liver to produce acute phase proteins important for killing pathogens. We also went through some of the important adaptive immune cytokines, many of which you heard of in earlier and will hear about in later lectures. For our final objective, we briefly introduced interferons. We know that there are actually 3 different classes of interferons, though we discussed mostly just type 1 and type 2 today. We learned that type 1 interferons play an important role in defending against viral infection by particularly through the production of interferons by plasmacytoid dendritic cells. We learned that type 1 interferons signal through a JAK-STAT signaling pathway similar to the other cytokines, and that the downstream result of JAK-STAT signaling is the production of hundreds of interferon-stimulated genes whose primary goal is to interfere with viral replication. And finally, we learned that type 2 interferons have many functions, including the activation of macrophages to become more inflammatory and better at killing and invading pathogens. That's all I have today, but thank you for listening to this lecture, and I hope it elucidated some of the earlier lectures in terms of the different roles of these cytokines and chemokines in organizing and mounting an immune response.